The second question comes from Kyla Hingwing, who asks, is Cas9 mRNA not being delivered, or is it not being expressed?
So again, a great question, and that's something that we're trying to tease apart, because while we don't see protein expression, the question of delivery to the cell and expression is not exactly the same thing. My hypothesis is that it's probably a combination of both of those factors. Encapsulation in an LNP of both the mRNA along with the guide is a little bit more complicated than just a single type of molecule. So it's possible that although the overall encapsulation efficiency of our formulations was quite good, that there are either some empty particles or there is some issue with the actual delivery of that mRNA to neurons. Cas9 mRNA is also significantly larger than the luciferase reporter we use. It's almost 50% larger. So translation from those larger mRNAs is less efficient, and particularly in neurons, which are not quiescent, but relatively so compared to dividing cells. It might just be that the translational activity for those longer mRNAs is lower than we anticipated.
And Sarah, maybe you can address, I know you did the experiments, the fact that the mRNA has the potential to create the protein. So you did some experiments to prove that the mRNA was functional.
Yes, we did use a commercial transfection reagent to deliver the same mRNA that we formulated in LNPs to a dividing cell line, just to make sure that there was still activity with that nucleic acid. And we did get Cas9 protein expression in a dividing cell line using that approach. So the mRNA itself in these systems has the potential to be translated into protein.
So my research group at the Center for Molecular Medicine and Therapeutics at UBC is focused on developing novel therapeutics for genetic brain diseases. And today we'll be talking about some of our work using lipid nanoparticles to deliver CRISPR genome editing. And I'll be giving the brief introduction, and then my really stellar graduate student, Sarah Thompson, will be carrying you through the data that's been supported by NMIN to date. So just some general introductions. Brain diseases represent a very significant unmet medical need. The brain is really hard to get therapeutics into, and the diseases can be quite complex. It also represents a massive cost to the Canadian healthcare system. In our lab, we study both neurodevelopmental disorders and neurodegenerative disorders. They can be familial or sporadic, caused by genetic diseases, genetic causes, or environmental, or in all cases, a combination of both. But for the sake of today's talk, we're going to focus mostly on just purely genetic disorders that have a clear monogenetic cause of the disease. And we can kind of break down these genetic brain diseases into having two underlying mechanisms. Mutations can cause a gain of function, which is basically a novel toxic species that is produced by the mutation, or the mutation can cause the loss of function and the loss of a normal protein. And this leads to very direct approaches to therapy. If we have a gain of function mutation, such as we have in Huntington's disease, you want to eliminate or remove that toxic or misfolded protein. And for loss of function mutations, we need to either repair or replace and increase the production of the normal protein. So just with that very simplified background, we've been focusing really on two different disorders in this program, both of which represent those two different models of disease mechanism. Huntington's disease, which is caused by mutations in the Huntington gene, specifically it's a expanded CAG repeat expansion within exon 1 of the Huntington gene, and frontotemporal dementia caused by progranulin mutations in the granulin gene. Even a single mutation leading to loss of function cause an age-dependent dementia called frontotemporal dementia. So again, Huntington's disease is a primary example of a toxic gain of function disorder, whereas frontotemporal dementia is a hemizygous null situation which is essentially a loss of function of progranulin. They affect different areas of the brain, but again, we've got to target the neurons in the brain to have effective therapies. And again, with a gain of function mutation, we want to reduce that toxic protein. And with a loss of function, we want to either replace or repair the gene to increase the production of the normal protein. Our goals in today's talk are to explain some of the advantages of how LNPs may help us to deliver nucleic acid therapies to the brain. We've set out to improve some of the LNP formulations through screening in primary neurons, both for siRNA approaches as well as for mRNA approaches, and we'll pass on some of the understandings we've gained from those studies. And ultimately, we wanted to apply these improved formulations to the challenge of LNP-mediated genome editing, a much higher bar for therapeutic delivery, and Sarah will take you through those studies. So when we think about current delivery of nucleic acid therapeutics to the brain, there are really two approaches that are currently in clinical stage development. Antisense oligonucleotides, an area that I've worked in both in the lab and clinically. In fact, we did the first ASO therapies for Huntington's disease in our clinic at UBC. These can be delivered directly to the CNS. They don't require any external carrier. They need to have repeat delivery because they're transient. Although they may have long-term effects, they aren't permanent. There are safety concerns, and these safety concerns have actually become more prominent over time with both the possibility of off-target effects, but also toxicity related to the underlying chemistry of the ASOs. They're very very applicable to gene knockdown approaches, which is why they've advanced in Huntington's disease. Less appropriate for gene replacement, although they can sometimes alter splicing with certain diseases, but primarily used as gene knockdown approaches. AAV, well known as a means of delivering nucleic acids to various tissues. They, again, require a viral carrier for the nucleic acids. In this case, they do have long-term effects. That can be good and bad if they're safe. But there also are significant safety concerns, and unfortunately, many recent deaths in current AAV clinical trials. They can deliver siRNA as knockdown or mRNA as gene replacement. But what we're really going to focus on today, and I think is of interest to this group, are our efforts using lipid nanoparticle delivery of nucleic acids to the brain. There's some really significant advantages, the primary one being how safe they are. They can be repeat dosed, unlike AAV. The effect can be transient, but also, again, we can deliver either mRNA for gene replacement, siRNA for knockdown, or a combination of guide RNAs and mRNAs for genome editing, which is where we'll finish up our talk today. Don't have to tell this group, there's been some really significant advances, and certainly the development and fast tracking of the SARS-CoV-2 LNP vaccines by Pfizer and Moderna has important implications for more complex LNP therapies. This slide is clearly out of date. It's from a review from 2021, and at that time there were about 30, and I understand from our most recent admin talks that there's over 100 LNP-based mRNA and nucleic acid therapies in clinical development. So a really incredibly rapidly developing field. Of import to this particular presentation, there was a really exciting publication recently in the New England Journal. This represents the first in vivo evidence of clinically effective and safe genome editing. This was a CRISPR-Cas9 delivery by LNP of the Cas9 mRNA and guide for a liver disease, transthyretin amyloidosis. So really exciting advances. And that brings us back to some of our very early work. So really based on some important first principle work done by Peter Cullis and the McVicker lab that showed that LNPs actually were effective in effective in delivering nucleic acids therapies to brain cells. We started working with Peter many years ago, and we're really impressed with the ability of nucleic acids to deliver LNPs to deliver nucleic acids to the brain. This is sort of our workhorse system. It's a primary neurons from mouse in a dish. These are non-dividing post-mitotic neurons, either from cortex or striatum. And here we just delivered an siRNA targeting Huntington, the gene that causes Huntington's disease, and we're able to show very effective knockdown in ex vivo neuronal cultures. We took those same LNPs and delivered them into the region of the brain, the striatum that is primarily affected in Huntington's disease, and we're able to show a significant knockdown. This was really exciting early data, but really we hadn't optimized any of the LNPs for brain delivery, so we set out, funded by NMIN, to do just that. And we knew at that time that LNPs were safe and effective with peripheral administration, but there really wasn't any data on what happened when we delivered them to the brain. We had this early data showing that neurons could be transfected by LNPs and that it could be done safely and effectively. And the goal of our initial NMIN program was to optimize LNP formulations for the delivery of genetic therapy payloads, with the ultimate goal to deliver these therapies to the brain. We focused on siRNA for a gene knockdown and mRNA for gene replacement. And I'm going to pass the baton over to Sarah Thompson, since she's actually done all of the work and is the best person to describe how we've used LNPs to deliver nucleic acid therapies to the brain. Sarah, take it away.
So thanks very much for the introduction. And again, I'd like to thank NMIN for organizing this and for encouraging my participation in our lecture today. Blair has already showed some early data from our lab using LNPs, both in ex vivo systems as well as in vivo. But I'd like to start my portion of our talk today and just take a step back to review why we and the neuroscience and nanomedicine fields in general became interested in the possibility of using LNPs in the brain. So the functionality of LNPs relies on two essential physiological factors. The first is the presence of apolipoprotein E, or ApoE, and also the expression of low-density lipoprotein receptors by cells that we're targeting with LNPs. So when the particles enter circulation, the outer-facing lipids in the particles can interact with lipoproteins in serum, which includes ApoE. And these lipoproteins adsorb to LNP surfaces, and when the particles reach target cells, those adsorbed proteins bind with LDLR receptors, which causes endocytosis and allows delivery of nucleic acid into the cytoplasm. We became interested in using LNP technology in the brain because we knew that both of these essential components also exist in the central nervous system. ApoE happens to be the most abundant lipoprotein in CSF, and LDLR receptors are highly expressed by neurons as a component of the brain cholesterol biosynthesis pathway. And as Blair mentioned, we definitely weren't the first people to come up with this idea. And in particular at UBC, this prospect was really nicely investigated by Peter's group as well as Brian McVicker's group. This figure is adapted from a paper that came out of the McVicker lab, and the panels on the left show ex vivo neurons in the top left stained with DAPI, as well as neurons in a brain section that are stained with live cell dye. LNPs that were used to treat these cells, both ex vivo and in vivo, are shown in the center with a red lipid dye. And then the merged panels on the right just show really nice cellular uptake in both of these systems. These cells were treated with an siRNA formulation, and we also know that the siRNA was functional and lowered expression of PTEN, which was the target gene in the brain parenchyma, so in vivo, compared to both uninjected and the non-targeting siRNA controls. As Blair mentioned, we also had worked with Peter for many years, and we were interested in building on this foundational work. So we designed a project that would improve the efficacy of LNP formulations for neuronal delivery. So in order to adapt LNPs for this purpose, we set out and designed an ex vivo screening strategy to identify the best combinations of lipids in LNP formulations for nucleic acid delivery. We isolated neurons from the brains of mouse embryos and treated them with LNPs after the neurons reached maturity at day in vitro 7. And to identify effective combinations of lipids for neuronal delivery, we manipulated 2 of the 4 lipid components in a typical LNP system. So we focused on ionizable cationic lipids, which promote encapsulation of nucleic acid and also allow for release from the endosome following endocytosis. And we also looked at different phospholipids, which can modify delivery efficiency. And the ultimate goal of this project was to land on an optimized formulation that worked really well to deliver our nucleic acid payloads. We tested formulations, again, of both siRNA and mRNA, and we did all of these experiments in reporter systems just to avoid disrupting endogenous gene expression. Formulations of siRNA were screened in primary neurons that we isolated from transgenic GFP mice. And after treating those neurons with LNPs containing GFP siRNA, we measured knockdown using qPCR. And then for mRNA, we used formulations containing luciferase reporter mRNA in wild type primary neurons, and we quantified luminescence after treatment. Our first screen was with formulations of LNP siRNA, and we compared 2 ionizable cationic lipids, D-lin MC3 DMA, as well as DODMA. And these formulations were prepared using disteryl phosphatidylcholine, or DSPC, as the phospholipid. Importantly, one of these combinations, MC3 and DSPC, is the same lipid combination that is used in the FDA-approved LNP siRNA therapeutic Onpattro. And this is a really common benchmark in the field when doing formulation screening. So in all of the subsequent experiments I show, this MC3 DSPC formulation is our benchmark control. In this study, both of our formulations achieved significant GFP knockdown compared to both untreated and scramble conditions. And we saw that the MC3 formulation was significantly more potent than the DODMA formulation at an equivalent dose. We then moved on to testing the performance of each of the ionizable lipids in combination with 3 additional phospholipids with unique chemistry. So we tested DOPC, DOPE, and DOPG. And what we saw was that each of these formulations had a significantly different effect on GFP expression and knockdown. So when prepared with MC3, we saw that the DOPG formulation was the most potent. And then when we did the same screen with DODMA, we saw the same trend. So based on this, we conclude that for siRNA delivery to primary neurons, DOPG formulations are more potent than formulations prepared with other phospholipids. We then went through the same type of screening protocol using systems with mRNA. And we started with the same 2 ionizable cationic lipids, MC3 and DODMA, formulated with DSPC. But here, unlike with the siRNA formulations, we actually saw that the DODMA formulation at the same dose was much more effective for mRNA delivery than MC3. We also completed the same phospholipid screen again, beginning with formulations of MC3. And in this case, the trend that we saw for siRNA delivery was maintained here. So the formulation containing DOPG was the most potent. But when we did this screen with formulations containing DODMA, we saw a different trend. So in this case, it was actually the DOPE formulation that was the most effective. And here we basically see that the best phospholipid for mRNA delivery actually depends on the type of ionizable cationic lipid that's present in the formulation. So compared to our LNP formulation that we used as a benchmark, the MC3 DSPC control, this project has been able to improve neuronal delivery of siRNA about 1.7-fold and of mRNA over 14-fold. We're really excited about these results and the potential of these formulations to enable some ex vivo proof of concept studies in neurons, which are a notoriously difficult cell type to work with and transfect. But we also became really interested in that higher bar of using these systems to attempt to gene edit in neurons. So that was the question we asked. We wanted to see if we could use these formulations that really effectively deliver payloads to neurons to enable gene editing. So to tackle this, we set out to deliver the components of the CRISPR-Cas9 gene editing system using LNPs. Just a quick review of CRISPR gene editing technology. It's comprised of two primary components, the Cas9 endonuclease protein, this gray protein shown on the left here, which cuts DNA, as well as the guide RNA, which directs that Cas9 endonuclease to cut at a specific locus dictated by its target sequence. The Cas9 protein and guide RNA come together in the cell to form a ribonucleoprotein complex, which can then cut and edit genomic DNA in cells at that guide RNA specified location. So to begin testing whether our optimized LNP formulations could improve gene editing outcomes in neurons, we designed this CRISPR experiment. And for this work, we cultured primary neurons from brains of transgenic mice harboring the human progranulin gene. As Blair mentioned, mutations in this gene are associated with some forms of frontotemporal dementia, so therapeutic correction is of really significant interest to us and the field. And to edit the progranulin gene in neurons, we used our optimized mRNA formulation to deliver mRNA encoding that Cas9 endonuclease, so the LNP formulation containing DODMA and DOPE. And we used the optimized MC3 DOPG formulation to deliver the guide RNA targeting the progranulin gene. We also used the MC3 DSPC formulation as a control to deliver both of the components. And in some cases, we also incorporated a non-targeting scramble guide RNA to assess the specificity of on-target progranulin editing. There are also a few additional experimental design factors that are important to include in any CRISPR-Cas9 experiment that can influence gene editing outcomes. And we included several of these aspects in our experiments. Most notably, the ones that I will be focusing on are the ratio of Cas9 mRNA to guide present during cell treatments, the overall reagent dose that we're using, as well as the duration of the exposure of cells to these reagents. And in all of our experiments, our primary outcome measure was the degree of on-target progranulin editing that we were able to detect, which was quantified using Sanger sequencing. And in some cases, we also measured neuronal viability because neurons are very sensitive to any type of exogenous stressor, particularly some lipid-based transfection reagents. So our first experiment compared 3 ratios of Cas9 mRNA to guide RNA at 2 Cas9 mRNA doses and at 2 time points. This plot shows those 2 time points, 24 hours shown in red and 48 hours shown in blue. And right now on the slide, we just see the results for delivery using our optimized formulations where DODMA DOPE LNPs were used to deliver mRNA and MC3 DOPG LNPs were used to deliver the guide. The degree of editing we achieved wasn't overwhelming here, and it was particularly underwhelming compared to the degree of editing that was achieved with this control MC3 DSPC formulation. So it was unfortunate here that we didn't see that these optimized formulations produced a better effect than our control. Overall, the efficiency of this approach was quite low. Across all formulations, we only measured about a 1.8% average on-target editing of the progranulin gene. And while the impact of LNP formulation in this case wasn't significant, there were some subtle differences in editing magnitude associated with some of these other experimental parameters that we were interested in looking at further. So for example, we saw that a 1-to-1 Cas9 mRNA to guide RNA ratio produced the best result in the largest number of conditions at both time points and across all formulations. We also saw that a larger Cas9 mRNA dose did appear to improve editing efficiency at 24 hours. And that increased exposure to reagents did not significantly improve editing. So in this case, the shorter time frame of exposure was probably the best option for future studies. And while these results weren't particularly encouraging, they were still informative for a first experiment. So we used these different observations to ask if we could improve the results. So we designed a follow-up experiment using sort of the best parameters that we were able to identify from this pilot, which consisted of a 1-to-1 Cas9 mRNA to guide ratio. We decided to increase the Cas9 mRNA dose to 3 micrograms per milliliter from 1. And we also collected neurons at 24 hours following treatment. And in this case, we also included a treatment condition with a non-targeting guide as an additional control. But we didn't see these parameters positively impact our results. So on this slide, the optimized formulations are shown here in green. And we didn't see any improvement in editing compared to the control, which is shown in gray. And additionally, we also compared on-target editing compared to that scramble in this experiment. And we saw that we weren't actually able to induce any significant on-target progranulin editing. Because viability is also important, we decided to assess that in this experiment. And we did see that not only did different LNP formulations have a different impact on viability, but there was also a really significant amount of cell death that was happening in these experiments. So at this point, we decided to take a step back and just reassess our approach. And we thought that because we had used separate LNP formulations to deliver the 2 components of the CRISPR system, we thought that perhaps transfection efficiency or delivery timing of those formulations was too low to achieve effective editing. So to address this, we decided to take an alternative approach where we formulated Cas9 mRNA and guide within a single LNP. So using this strategy and an identical experimental design as the one from the first experiment I've presented today, we actually did see a modest improvement in editing outcomes with the co-formulation. We tested both optimized formulations to deliver both components, so DODMA DOPE as well as MC3 DOPG. And again, we compared the activity of both of those to the control. And once again, although we didn't see a significant improvement related to LNP composition, there was a very clear effect of time. So in this case, the degree of editing more than doubled between 24 and 48 hours. Using this co-formulation strategy at the 48-hour time point, we also noticed that a 1-to-4 ratio of Cas9 mRNA to guide, so in excess of guide, was more effective than in our previous studies. And similarly to our first attempt using separate formulations, it did seem that a higher Cas9 dose was associated with a more significant editing effect. Once again, we also decided to look at neuronal viability because this was important in our previous experiments. So we did a follow-up study to assess viability using the set of conditions that generated the most significant editing results from this co-formulation work. And the results of this experiment actually indicated that cell death was a very significant factor. So it was less significant for our optimized formulations, which was positive news, but more than 50% of the cells were dying in all of our treatment conditions. And at this point, we got concerned that the gene editing we measured could have potentially been an artifact of cell death rather than genuine on-target editing. So in light of this, we once again reevaluated our approach and decided to measure progranulin editing at lower doses where we could preserve neuronal viability and remove that as a confounding factor. So we used the same optimized treatment parameters identified from the last experiment, so that 1-to-4 Cas9 to guide ratio and a 48-hour time point. But in this case, we lowered the Cas9 mRNA dose up to 10-fold. This plot shows the viability of those cells at 2 lower Cas9 mRNA doses. And here, once again, we saw a unique impact of formulation on neuronal viability, but we also saw that we did choose doses where we were able to almost entirely preserve neuronal viability at least one treatment, which in this case was 0.03 microgram per milliliter mRNA. So we were excited about this possibility and used these treatment parameters to assess on-target progranulin editing. Unfortunately, the magnitude of progranulin editing that we measured using these parameters didn't improve editing at either of the doses, although we did measure a statistically significant signal using one of our optimized formulations, DODMA DOPE. So in this case, it looked like we were able to achieve around 1% on-target progranulin editing as compared to that off-target scramble condition. Although not overwhelming, this was a good step in the right direction. And although we'd done a lot of work at this point and made some progress, we were also still curious about why we weren't able to achieve more significant editing, because we were able to elucidate some factors that could contribute to other aspects of this system working, but the overall magnitude of editing that we measured was still quite low. And because our strategy involves Cas9 delivery in the form of mRNA, we decided to do a series of Western blots to look for Cas9 protein to see if the mRNA was being translated in the neurons that we were treating with LNPs. So we blotted for Cas9, which is shown on these blots in green, and just compared to a loading control shown in red. And our positive control for these blots was actually Cas9 protein, a ribonucleoprotein provided to us from some of our collaborators that was spiked into lysate from untreated neurons. In this case, although the Cas9 protein that we spiked in gave us a nice signal, we did see that protein was not detectable using this method in our LNP-treated samples using any of the formulations that we set out to test. So while this won't immediately help us improve gene editing efficiency, this experiment was still important for this work just because it does indicate a potential issue with Cas9 mRNA delivery. And it shows that while we were able to significantly optimize delivery of simpler cargoes like siRNA and mRNA alone, there are other factors that make a gene editing application more complex in any system, but perhaps particularly in neurons. So improving delivery of Cas9 mRNA is the focus of our current and upcoming work. We've learned a few important things that will inform the future work of our lab as well as others from all of these experiments. We show from our screening work that LNP composition can significantly impact formulation efficacy. We also showed that systems optimized for siRNA delivery are not optimal for mRNA delivery to neurons, which is something that we didn't expect and important for any type of future experimental design. We also know that neuronal viability is of critical importance for data that is interpretable for these experiments. And for genome editing, we did see that mRNA and guide RNA co-formulation may be slightly more efficient than using separate LNP formulations to deliver CRISPR gene editing components. Our future work is primarily focused on improving LNP-mediated delivery of Cas9 while preserving neuronal viability. And then once we are able to achieve good delivery in healthy cells, we will return to evaluating the impact of LNP formulation type on editing efficiency. This work was done largely in the Levitt lab, but it was absolutely a team effort and supported by our collaborators, both other group members as well as collaborators at UBC. I'd like to thank Dr. Terry Petkow, formerly of the Levitt lab, for supporting all of this work. as well as Jay and Jerry from Peter's group for making a lot of the formulations we tested, and Dom from Peter's group as well for lots of helpful discussions in the early experimental design of these experiments. I'd also like to thank our collaborators and funding sources, including NMIN, that made it possible for us to do this work. And I'd now like to thank you all for your attention and open the floor to any questions you may have for Blair and myself. Thank you.
So the first question is from Tina Chen, who asks, why do you think DOPG was the most potent?
I can take that question. At least for siRNA delivery, it was most potent, and that phospholipid does have a negative charge in it. So that could potentially improve the delivery of the small siRNA. It does become a little bit more complex when we ask the question of why DOPG wasn't the most potent with mRNA, but particle size is also an important characteristic to consider. And in this case, the DOPG particles were also slightly larger, which means that more material can be delivered per endocytic event. So that's likely also a portion of why we saw a better effect there.
I see. The second question comes from Kyla Hingwing, who asks, is Cas9 mRNA not being delivered, or is it not being expressed?
So again, a great question, and that's something that we're trying to tease apart, because while we don't see protein expression, the question of delivery to the cell and expression is not exactly the same thing. My hypothesis is that it's probably a combination of both of those factors. Encapsulation in an LNP of both the mRNA along with the guide is a little bit more complicated than just a single type of molecule. So it's possible that although the overall encapsulation efficiency of our formulations was quite good, that there are either some empty particles or there is some issue with the actual delivery of that mRNA to neurons. Cas9 mRNA is also significantly larger than the luciferase reporter we use. It's almost 50% larger. So translation from those larger mRNAs is less efficient, and particularly in neurons, which are not quiescent, but relatively so compared to dividing cells. It might just be that the translational activity for those longer mRNAs is lower than we anticipated.
And Sarah, maybe you can address, I know you did the experiments, the fact that the mRNA has the potential to create the protein. So you did some experiments to prove that the mRNA was functional.
Yes, we did use a commercial transfection reagent to deliver the same mRNA that we formulated in LNPs to a dividing cell line, just to make sure that there was still activity with that nucleic acid. And we did get Cas9 protein expression in a dividing cell line using that approach. So the mRNA itself in these systems has the potential to be translated into protein.
And that actually is also answer. I think there's a question about using lipofectamine. And unfortunately, we can't use lipofectamine in primary neurons. These are very difficult to transfect cells, and standard transfection techniques just end up killing all the cells. So in order to prove that the mRNA was working, we did use a commercial reagent, but we used it in HEC293 cells, cell type in which you can. So that's the answer to why we can't use lipofectamine in primary neurons as well.
Hi, Sarah. That was really great. In retrospect to the end of your talk, what was going on when you saw that difference between waiting 24 and 48 hours?
So the difference between that experiment and the other experiment that I presented was, first of all, the LNPs were two separate formulations to deliver the cargos versus co-formulation. And I think that the reason we did see the slight increase only in the case of co-formulation is that we were measuring some on-target editing. And one of the challenges between translating these reporter systems into a more complex approach like this is that luciferase mRNA in particular is translated very quickly, and you can see a reliable luciferase signal even after 4 hours of treatment. So in the case of gene editing, we need the mRNA to be translated into protein. Then that needs to associate with a guide to form a ribonucleoprotein complex translocated into the nucleus, and then editing at that site has to occur, which is obviously a more complex process than just translation of a protein in the cytoplasm. So barring that confounding factor of toxicity, I think what's happening with that time point is that increased exposure to those reagents is actually just giving them time to have their intended editing effect.
Thank you.
So then Xiaoming Zhu was asking, have you tested your new formulations in vivo in the brain? In vitro situations might be quite different from in vivo. Thanks.
So I'm happy to answer this one, Sarah. Give you a bit of a break. So absolutely, this was the next step in our experiments, and this was an aspect of the program that was carried forward by a very talented former postdoc and research associate, Terry Petkow, as Sarah mentioned. And we have looked at the in vivo delivery of the luciferase mRNA and the primarily the siRNA. And I think to summarize, we did see some improvements in those experiments, but we haven't got to a point where we feel confident moving forward with the more complex genome editing experiments. So the goal is ultimately to bring those in vivo, but much of the work Sarah's presented today hasn't been taken in vivo yet.
Great, thanks for that. Then there's a question from Wenchen Zhao, who asks, how much PEG is present in these formulations? PEG can often cause toxicity in the in vitro work.
Yeah, that's definitely true. 1% PEG is something we used for all of these formulations. And we actually decided to fix the molar ratio of the composition when we were doing screening, just to avoid basically creating an incredibly complex matrix of treatments to test for the screening work. So all of these formulations are 50% ionizable cationic lipid, 39% cholesterol, 10% helper lipid or phospholipid, and 1% PEG. We have gone up to at least 3% PEG with neurons, I believe, and we haven't seen a significant effect on neuronal health. It's largely the nucleic acid dose and the overall lipid dose that we see has really significant effects on viability.
Thanks. Yasmin asks, do you think the complex niche microenvironment of the brain plays a role in hindering the targeting of the formulations? Consider perhaps secretions or genes that interact with the formulations.
It's a great question, Yasmin. I don't know that we can really make a lot of comments with all of the formulations and the more recent work, which has all been ex vivo. So in the ex vivo system, we're isolating the primary neurons and really just looking at, once you've delivered the LNP to the neuron, looking at uptake and expression within the cell. Our experience to date has been, when we have efficient and effective delivery to neurons ex vivo in that system, and we do direct delivery into the target region, there is similar levels of uptake and expression. So it's a really interesting question. The brain is very complex, and the microenvironment is, and obviously in our primary neuron cultures, we only have a single cell type. We don't have astrocytes, microglia, oligodendrocytes, other cell types. which might influence and certainly uptake the LNPs. But when we've looked at LNP delivery in vivo, we see similar levels of uptake that we see. So I don't know, and we can't answer the question. But as we move forward, we'll certainly get more information on that. My simplistic answer is we don't see that the microenvironment is altering the efficiency significantly, and what seems to work in primary neurons seems to work with direct delivery in vivo.
Two more questions from Tina Chen. Why do you think delivering Cas9 affected neuronal viability, and how do you plan to preserve neuronal viability in the future?
This is a great question. And it really, I think, comes down to the ratios that we chose to deliver Cas9 and guide together. So what we did to make these experiments consistent was we calculated all of our dosing based on the amount of Cas9 mRNA that was present. But in order to then deliver various different amounts of guide with that constant Cas9, there's a lot of difference in the amount of lipid that is present in those formulations for the same dose of mRNA. So an excess of guide requires more lipid to encapsulate as compared to less. And what we see, especially with the formulations we used, where there was an excess of guide in that 1-to-4 pairing, viability was a significant concern. So one of the things that I'm interested in testing is going back to reformulate and actually altering that ratio where mRNA is in excess rather than guide, to try to maximize the amount of Cas9 expression we get while there's still enough guide to enable targeting. That means we can deliver both components with significantly less lipid, which I think will really help the neuronal health aspect of these experiments.
And maybe a follow-up that helps to answer the next question that Tina raised, which is, you know, we had extensive experience delivering siRNA, you know, for many years, and I'll take a step back. We have in the lab really tested every possible transfection technique in these primary neuronal culture systems. We've looked at all the various electroporation and viral mediated delivery, as well as all the commercial reagents. And we've never seen anything that was as effective or as well tolerated as LNP delivery. And again, most of that work was done with small siRNAs. But we actually were really, I think the presentation probably undersells us. We were really surprised at the levels of toxicity we were getting and the complexity when you go from a small siRNA now to more complex formulations that include an mRNA and an siRNA. And you know, as you increase the complexity of your cargo, the number of parameters that can change and do change, and the number of parameters that Sarah's tested go up really dramatically. And you know, we were actually really shocked when we get to these more complex systems that we started to have these problems with viability. We've worked for many years with siRNA and really see almost no, we've seen very high levels of targeting with very low levels of toxicity and very high viability, and saw that translate very nicely in vivo. So one of the things that is a take home is that as you move to more complex cargoes, things get more complicated. So we were absolutely convinced, and are still absolutely convinced, that LNP delivery to the brain is a safe delivery system, and it works very well with certainly simple cargoes. But we're finding that when we move to these more complex systems, it's taking more work and more effort. But I think we'll get there.
Can I ask a sort of a related question, which is, so we've been playing with different, you know, lengths of nucleic acids that we load into lipid nanoparticles, and efficiencies are quite affected by the structure that or can be affected by the structure that is adopted by the nucleic acid inside. So I was just curious to know, when you do your loading efficiencies, did you have equal, you sometimes double loaded nucleic acids in one nanoparticle, sometimes in individual ones into separate ones. Did you notice either variations in, so did you get loading efficiencies that were, I didn't even know what your values were. Maybe you quoted them, but I didn't see them. I don't remember hearing them. Were they different on the three circumstances? One type, the second type, or both together into the same LNP?
Right. So we measure encapsulation efficiency for all of these formulations. I didn't show that data just for time today, but all of them were at least 85% or higher. And I think the general benchmark is 80% or higher is considered sufficient. It was very good encapsulation, both for the case of the separate particles as well as in the case of co-formulation. Mm-hmm. And I have seen some data, like you said, that shows with increased mRNA size or perhaps secondary structure that there is less efficient encapsulation. We haven't seen that here, but it's possible that there is something else going on with the physical structure of the particles that means this system has been more difficult to adopt.
Yeah, I suspect some of the things that are intended to inhibit endonucleases may be at play sometimes, but that's very interesting. And then one question, if I might, the efficiency was higher when the two nucleic acid cargoes were carried together. Could you just, or as I recall, could you just tell me why you thought that was true? Did it have to do with the timing of the why do you think that was better?
So we have done a little bit of work on working out transfection efficiency of different formulations in neurons, also ex vivo. And transfection efficiency is reasonable and sort of compared to what you would see with a commercial reagent, but it's also not 100%. And I think delivering guide RNA in one formulation type and Cas9 mRNA in another means that fewer cells are both going to endocytose both types of particles and actually end up or produce the result of Cas9 protein and guide RNA being present in the cytoplasm to form the complex that's required for editing than if you deliver both of those molecules together. Because when they're separate, only a subset of those cells is going to take up mRNA particles and only a subset will take up the guide particles. But there's nothing to say that those populations of cells are the same. So at least with the co-formulation experiment, we know that if we are getting endocytosis, we are delivering both types of nucleic acid simultaneously.
Yeah, right.
Yeah, maybe Gilbert, to follow up on your question, it may have been somewhat simplistic for us to think that all mRNAs are going to be the same, and I think the small siRNAs, because they're very much more consistent in their size and length, probably a single formulation will probably be broadly applicable. But I think one of the unfortunate take-homes from this is that likely different mRNAs may have different optimized formulations, and we've optimized a GFP mRNA, and that may not be this optimal formulation for Cas9, and it may not be for other mRNAs based on size and subsequent secondary structure. So that I think is an unfortunate take home. And I guess the other thing I would say is we do need to be careful about over-interpreting too much. I mean, we do think that the data was supportive, that the co-delivery was probably better, but the levels of editing efficiency are so low that and perhaps in some of the experiments there was none, and the expression of Cas9 was low and undetectable, that I don't want to, I think we should be cautious in over-interpreting any of the data. But I think Sarah's done a great job of gleaning what we can from the experimental data that's there, but we don't want to over-interpret things.
Yeah, no, I agree. The caution I agree with, but I also agree with the excellence of the results in terms of the quality of what's being analyzed. analyzed. There are a couple of questions here. From Natalia, the question is, what were zeta potentials of the two optimized formulations? Do you know?
We actually didn't measure zeta potential of these formulations, but it's not the first time I've had that question. And it's definitely something we'll look into because it is important.
Right. The last question, or the second to last question on the chat is, what about a different ionizable lipid? And then they describe Moderna, the SM102, and the MC3. And it has to do with tolerance. I guess the answer is there are a lot of places you could go with different formulations. Is a sort of a generalization I might make. And do you want to add to that, Sarah? Any comments? What other formulation might you imagine trying?
Sure. I will say that for the sake of this presentation, the data was condensed to what was most easily discussed. We have tested more formulations and more ionizable lipids than I've shown here. It is a good point that there are some new lipids, including like ALC0315, which is in the BioNTech COVID vaccine, that are incredibly potent that weren't available to us when we started this program. And it would definitely be probably worthwhile to test some of those new lipids if we could get our hands on them. I think I would revise the experimental design a little bit for this application specifically, but we could also incorporate them into a screening experiment. It just sort of depends like what we're interested in doing with the optimized formulations.
I think there's a gigantic horizon with a lot of exciting things to try. And I think you guys are on a good direction for that.